I want to thank organizers for inviting me and organizing this very interesting meeting. I'm going to talk about metabolic pathways that are affected by tafazine deficiency, and often it's necessary to disclose any financial interest. Nothing have to disclose. I wish I had, but that's the truth. Now, as we realize now, tafazine deficient mice are a great model for Barth syndrome, but as Barth syndrome itself, the phenotype in knockout mice is very complex, and there's many pathways are involved, and many organs are involved because it's not a tissue specific; it's a systemic knockout. On top of that, doxycycline also adds certain variations to the phenotype. So far, we don't have any tissue-specific knockouts, but once we have, maybe there will be a more clear picture. But it's clear that many different pathways are involved in this development of the pathophysiology of the Barth syndrome in these mice. And I will try to give this little background about phenotype of these mice and the mitochondrial defects, mostly in the heart. Skeletal muscle are removed because of short time. And the mitochondrial proteomics, I noticed there was a previous speaker also had some interesting data about it. Electron transport chain, and the attempt to make sense of mitochondrial proteomics with the bioinformatics and also supercomplexes. Now, tafazine knockout mice became available, I believe it was 2009, and many people giving me the credit for that. I have nothing to do this. I just was along with Barry, we were first lab who received it from Barth Syndrome Foundation, these mice, and it was done in Germany by a commercial company. And we just started breeding them, establishing colonies, and sharing with people. And it was paid by Barth Syndrome Foundation. And gene silencing is achieved with shRNA interfering, so it's not a regular knockout, it's just shRNA mediated. And it has this lack of tissue specificity because of how it's constructed. Now it's inducible system, which is nice because we can induce the knockout, and after this, discontinue dogs and see what happens if function comes back. Efficiency is more than 90%, and we all know that cardiolipin is, especially L4 cardiolipin, is dramatically reduced in these hearts and skeletal muscles. Mitochondrial abnormalities, structural abnormalities, and increased mitophagy is evident. And these mice develop left ventricular or biventricular dilation by 7 months of age. But cardiac defects are also by Colin Funn's lab being reported at the embryonic stage. And we initially missed that when we characterized, but I think it's because it's linked to the genetic background of these mice. So if I have time, I will talk a little bit. And they have impaired exercise endurance. And what means if we put these mice on the treadmill, they just run a little bit, but after this, it's a forced exercise. So it's stimulated by electrical shock. So, but if after we increase intensity of exercise by increasing treadmill speed, they prefer to receive electrical shock rather than run. So, of course, we remove immediately, but we can record if we give the intensity they can sustain. We can observe that initially these mice, they start increasing the oxygen consumption, they become very glycolytic, but after this, oxygen consumption goes down, so they are, they become more glycolytic, and but while wild-type mice, they have no problems to keep up running, and they increase oxygen consumption this rather dramatic way here. So plasma lactate, if we measure somewhere here, plasma lactate is higher much in tafazine knockout mice, and this is aerobic strength. If we give them grip strength, which is more anaerobic task, they don't have there's no difference between these two, tafazine knockout and wild-type mice, unless we repeatedly subject them to the grip test once again, then it becomes more aerobic, and they get tired, and you can see this difference then. In heart, when they become 7 months old, they develop cardiomyopathy, and the heart looks really messed up. So the mitochondria are smaller, fragmented. Sarcomeres are also in disarray, and this is probably from overproduction of reactive oxygen species. Same picture Bill saw in his nice model. But before they become old, we don't see much phenotype in EM2. But what we see, mitochondrial defects in the functional mitochondrial defects in young mice, or even in neonatal cardiomyocytes. If we take like a one-day-old pups and we isolate cardiomyocytes, we plate them in the glucose. This is important in the glucose, because if we get a different picture if we put a different sugars here. But when we put in the glucose, in the basal level, we don't see much difference between them. And but if we tax them with the FCCP, then it becomes really difference is dramatic, and you can see the respiration capacity or respiratory reserve is decreased by 40%, and that's when we uncouple them with the FCCP. Same time, glycolytic rates are in the basic level are higher. So it tells us that some kind of remodeling is going on here, the metabolic switches, and I'm going to talk about this phenomena. Now, we also attempted to, because in previous slides, this tells us there's a defect in electron transport chain, and we looked at electron transport chain complexes. Now, complex 1, we couldn't find any defects in activity, although the proteomic analysis showed differences. I'll show later. Complex 3 was dramatically reduced. Activity was complex 1 to 3 segment was decreased by 50%. Complex 4, cytochrome C oxidase, wasn't affected. And the complex 5, we had a different picture. This is the old data, which shows that 30 degrees, but if you switch this activity to 37, we have about 15%, 20% lower than in wild types, in tafazine knockout mice. And again, we measure this by actually not direct way, because it's ATP synthase, we measure here ATP hydrolysis, ATPase activity, so which is reverse reaction of the. So, but anyway, this is also probably is affected complex 5 for ATP synthase. So, but for primary target, we thought it was complex 3 because activity was reduced in most dramatic way in complex 3. Now, this is a cardiac phenotype. I've tried to make this different data to combine in this single slide. And this is Collins' paper, which shows this about fetal cardiac failure in tafazine knockout embryos at 12.5 days. And but about 50, and they die in uterus. But about 50% of transgenics, they are still born. And they develop without any significant defects until they become 7 months old, and they develop these biventricular dilations. And I'm interested what's going on here. So what makes heart to adopt these defects in mitochondrial respiratory chain and mitochondria in total. So what pathways are here upregulated or downregulated, and how these mitochondrial deficiencies are compensated. So if you remember yesterday, Dr. Jeffries talked about a little bit about cardiac shape, and this is the perfect example how what he meant. This is 4-month-old mice. There's no functional cardiac deficiency yet developed in these mice, but look at shapes. These mice have, wild-type mice have football kind of like of shape. This is left ventricle. And Deficiency knockout mice have more basketball kind of shape, and those are more round shape. And if you measure these to these, and you can see these changes are rather significant. And that's the shape of heart, this basketball kind of shape of heart is positively associated with left ventricular non-compaction and dilated cardiomyopathy later. So I want to talk about a little bit about metabolic shift. What I mean is that during the embryonic development in a relatively hypoxic environment, the heart utilizes anaerobic glycolysis as a main source of energy, whereas in adult cardiomyocytes utilize oxygen-dependent mitochondrial oxidative phosphorylation as main energy source. And in mammals, metabolic shift occurs when they're born and in early postnatal life, when they switch to the more fat-containing diet as milk, breast milk, and they got exposed to the oxygen. However, if system is broken and the mitochondria are not properly function, this metabolic shift can go wrong, and it doesn't go all the way to completely shift from one type of metabolism to another. And we hypothesize that mitochondrial defects in Deficiency knockout heart prevent the full metabolic remodeling at birth, causing the heart to rely at least partially on less efficient alternative metabolic fuels such as amino acids, carbohydrates. And the fetal energy metabolism allows heart to adapt to the mitochondrial deficiency and fulfill energy requirements at basal physiological conditions, but is detrimental in the long run or under stress conditions. And the first thing we did a gene expression array. So we compared adult 3-month-old mice gene expression patterns in wild types and the knockouts, and see what's the difference between them. And we, rather to giving to just a list of genes, I combined them, or software combined them into the pathways. Those are upregulated and those are downregulated in these hearts. And this is mRNA-based experiment. So this is not protein, this is mRNA, which is not necessarily translated to proteins, but it tells us something. And we saw, huge upregulation of the folate metabolism, and one carbon folate coupled to one carbon metabolism in these mice. And these colors actually, they show these differences. So the more different the colors are, it's more different is the regulation of, the level of these mRNAs. And Deficiency itself somewhere here. So we had about 7 to 10-fold decreased Deficiency gene, while upregulation, just to have a flavor, it's 25 or even in some cases is 1,000-fold. And it's not because of this folate metabolism is so much active. It just, initially, in adult heart, there's not much folate metabolism. It's very active in the fetal heart, or in cancer cell, hypoxic heart. Even it's associated with cardiomyopathy, but in normal adult heart, it's mostly silent. But all these genes are upregulated in this tafazine knockout hearts. Amino acid synthesis and transport, unfolded protein response. And downregulated genes are fatty acid metabolism genes, calcium homeostasis, branched amino acid catabolism, Krebs pathways, and others. And there's also signaling molecules, several signaling molecules that are hugely upregulated in these mice, but we don't know the actually role of these molecules. Basically, they are not supposed to be expressed in adult heart. Again, these data are done on the Affymetrix DNA array, but by the time we were done with this data, we saw that another group, Michael Kibish group, published RNA sequencing data on the same model. So, but data pretty much overlap, except these signaling molecules we couldn't find in those data. And data is available on the NCBI website. It's in the public domain, so anyone can go and analyze this. What we did differently, we tried to do proteomic approach, use proteomic approach, which is more down to the point because it's not RNA, it's measures the protein levels and the differences in these proteins. So we tried two different approaches, one two-dimensional gel-based and then another eye-track. I briefly will tell you this is basically two different samples from wild type and knockout we labeled with different fluorescent dyes, mix them, run them on the 2D gel first by isoelectrofocusing of this SDS page. And after this, we quantify the intensities of each spot and see how much green there, how much red is there, which one is increased or decreased. And we cut these spots and do the mass spec and analyze the and identify proteins, while this part is more direct, and we just labeled proteins with specific tags with non-radioactive stable isotopes, and after this we do the mass spec. Each has its own ups and downs, but I like this because this also tells you the post-translational changes because phosphorylations or glycolizations can be also be spotted here. But we use both methods, and we found that 2D gel gives you the more spots as expected because two-dimensional gel-based technique allows to identify post-translational modifications. So we identified 2,300 individual spots on this gel, and this was mitochondrial preps. It wasn't total heart. It was only mitochondrial preparations. 82 were altered spots, and after this we applied some, cutoff was 1.5-fold, so whatever was up and down 1.5-fold or more with the 95% accuracy, we only left those. From there, we identified these spots, and from 33, 30 were mitochondrial proteins, and the 24 non-redundant proteins. That means the several spots were actually the same proteins here. While here we were able only to get 533, and 5 mitochondrial proteins were satisfying these conditions, and eventually we got 24 here and 2 more here. So total 26 proteins were identified, and that is how this looks. It's first isoelectrofocusing was done in this direction, and after this SDS page at these individual spots, and those spots were that are changed one way or another in between tafazine knockout and wild type hearts. And from that we identified 33 which were really strongly changed and Okay, and this is how analysis looks. Those are individual spots, which sometimes we cannot detect even by eyes, but software can detect, and the volumes of these spots are compared. If there is consistently in the two or three samples, then we identify these spots. And those are the final data. It's just a list of proteins that came up in our screening, and you can see this. All mitochondrial complexes are decreased, except complex 4, actually, 1, 2, 3, 4, 3, and 5. The subunits of these complexes are decreased. Also, they decreased iron, sulfur, cluster assembly as an enzyme, which is upstream for complex 3. And actually, 2 complex 3 subunits are affected. Ubiquinone biosynthesis proteins, 2 of them, and the chaperone-like protein for BC1 complex, or complex 3 again. So those are down significantly. Interestingly, folate metabolism, which we identified in the RNA-based method, it came up again. So folate metabolism was manifold increased in this heart mitochondria. Mitochondrial processing, mitophilin, which is the member of this Micos complex, was early mentioned here, was all upregulated, and it's crystal junction protein. Myoglobin, old boring myoglobin, was also, but this mitochondria associated myoglobin. I'll talk a little bit about this, because myoglobin is the principal oxygen delivery system in the cell. So it takes oxygen from the bloodstream, and the oxygen becomes oxymyoglobin. It delivers to the mitochondria, releases oxygen there, and this system is works because from literature it's clear that there's no specific receptor for myoglobin. It's more electrostatic interaction with my membrane, and it looks like this mitochondrial associated myoglobin is decreased, while total myoglobin is not. Total cardiac myoglobin is the same, you can see here, but mitochondria associated is decreased. So I was wondering maybe it's just the affinity of myoglobin is decreased to mitochondrial membrane, and can deliver less oxygen to the oxidative phosphorylation system. And potentially it can explain some of the data which shows that blood of Barth patients, when they exercise, is oversaturated with the oxygen, but they are not able to extract this oxygen from the blood. Okay, those are the complexes, mitochondrial complexes, and the subunits that are affected or decreased in the tafazine knockouts are highlighted. This is NDFA5, which is not catalytic subunit, but it's important for the electron transport. As complex 2, you can see here, this is catalytic subunit. It is decreased almost twofold. 2 subunits of complex 3, Rieske and cytochrome C1, both are decreased, and they, just for reference, cardiolipin is part of the crystal structure of complex 3, and it's here. Complex 4 wasn't affected, and the catalytic subunit of ATP synthase was also down. And we tried to use the bioinformatics approach to make sense of this data. And we try to analyze the pathways that are affected by those changes in the proteome. Actually, every protein is involved in the multiple pathways, and they can affect multiple cellular processes. And we used DAVID, which is the database for annotation, visualization, discovery, and TopGene, which is maintained at Cincinnati Children's, and those databases are free, and the Ingenuity pathway analysis, which is commercial and very expensive, but I was I was lucky to hook up with a person who actually owns the license, and he analyzed data for me. But anyway, this analysis, because Ingenuity is so expensive, it has most interesting and nice phenotype. Yeah, I'm almost done. And you can see these pathways are the mitochondrial dysfunction and the folate metabolism, and the catalysis and the ubiquinol pathways are down, while folate metabolism and protein degradation is way up. And also, if you take a lipid disease-associated networks that also came up with in the software package, you can analyze them. You can see these processes that are most affected in this tafazine knockout hearts. And you also can see that not only those proteins that are affected, but proteins that they potentially interact. So I can see, but those are nodes where those proteins are centered, and apolipoprotein 1, adrenergic receptor beta 2, and the other proteins which needs to be analyzed how these processes are affected. And finally, I will talk about a little bit complex formation, super complex formation. And what we did, we first, we solubilized mitochondria, ran them on the gel, ran them on the centrifuge overnight, and separated these super complexes and run on the Western, and you can see super complexes are here. They come in the 25 and 20% of sucrose gradient. And in knockout, we can see the complex 4 is here. It comes in the same with complex 3 and with complex 1, but it's gone, they are gone in the tafazine knockout mice, so complex 4 is completely absent in the fraction where it's supposed to be in wild types. And because complexes are also interacting with this fatty acid oxidation system, it's been established, we thought, why we don't check on the same gel fatty acid oxidation systems, and instead of having the supercomplex, you may have a hypercomplex with fatty acid oxidation, and you can see this here. If you see these proteins here, those are fatty acid oxidation proteins, which are missing in the supercomplexes of in tafazine knockout mice. And this is final slide, which shows the supercomplex is assembled in the tafazine knockout heart, and the fatty acid oxidation is enzymes are also in part of the supercomplexes, and those are the dissociated supercomplexes in the tafazine knockout mice, and the fatty acid oxidation enzyme is also down. And this is my summarizing that complexes are altered and destabilized tafazine knockout mitochondria. Fatty acid oxidation system becomes disengaged. Myoglobin to cardiolipin depleted mitochondria is significantly impaired binding of this and the mitochondrial defects in tafazine knockout hearts induce activation of genes that were consistent with metabolic shift toward glycolysis, amino acid metabolism, and other pathways. And I want to thank my co-workers, Corey, Jan, and other people at Cincinnati Children's and Barth Syndrome Foundation, and was funded by Cincinnati Children's and NCBI, NIH. Thank you.
Okay, thanks, Zaza. I think we have time for 1 or 2 questions.
So that was very interesting, of course. I want to ask you that in addition to myoglobin, I think this kind of relatively new member of this globin family, cytoglobins, are involved in delivery of oxygen to mitochondria. Was it in your panel?
No, we didn't find that. Is it expressed in heart also?
Wow, it has different levels of expression, so I don't know how much of it, because it's at micromolar levels usually it's expressed.
Well, from 82 we only analyzed 33, which was highest change, and we didn't find among them.
So the reason I'm asking you is because cytoglobin, they are present in penta and hexa-coordinated forms, which is relevant to binding of oxygen. Mm-hmm. And so you can regulate binding of oxygen and delivery of oxygen by binding lipids, because this penta-hexa-coordinate transition is lipid dependent.
Well, we'll check it probably without proteomics, but just using antibodies or labeled proteins. Probably it will be the more direct way to check this if this affects the binding in the Barth mitochondria.
Another question?
Did you check the level of coenzyme Q?
No, we haven't checked. Also, upstream enzymes that make coenzyme Q were all down. So probably the coenzyme Q is down, and we actually tried to rescue with the coenzyme Q Drosophila model. It worked perfectly. We gave Drosophila, tafazine knockout Drosophila, coenzyme Q, and they start climbing much better than knockouts. And I didn't include it here, but that experiments were done, and it seems the coenzyme Q makes, at least in Drosophila model, real difference. They rescue climbing, they rescue ATP and the membrane potential.
Yeah, then probably did. So you showed the complex 1 is normal, but 1, 2, 3 is down, right? So
1, 3 is down.
1,
yeah.
3 down. So could it be just coenzyme Q, not the complex 3? Right?
Well, when we measured activity, coenzyme Q was in excess. Yes, we didn't rely on the internal coenzyme Q. We just add our coenzyme Q. Yes.